So our next speaker needs no introduction, but he's going to get one anyways. Ron Davis is probably one of the greatest out-of-the-box thinkers in the field of genetics and genomics. It's been a pleasure for those of us who have gotten to work with him over these years. And we were kind of playing a 6 degrees of separation thing the other day with some of my distant colleagues. And I think almost everybody who's good in genetics and genomics is about 6 degrees separated from Ron Davis. They've either kind of come through his lab as a postdoc, a PhD, or they've collaborated with him. He's really transformed the field in that way. He is also credited with being one of the greatest living inventors on a list that included Elon Musk. Timely. And Jeff Bezos, how about that? Yes. So Ron is, Ashley introduced him from a personal standpoint. I just want to impress upon you who are maybe outside the field of genetics just how much he's actually changed that field. He's credited with helping to launch the recombinant DNA revolution. That before there was CRISPR and gene editing, there was recombinant DNA, and there was just as much excitement then, and that had just as much of a transformative effect on what we're able to do as genetics researchers. The scientists who have gone through his lab are really some of the greatest in this field. And as you see, Ron's approach is really like, let's just try new things, let's break things, let's innovate. And he's inspired generations of us with this. I'm actually 2 generations removed from him. He's my PhD grandfather, and that's been a major influence on my scientific career. The idea that technology drives biology has come through, I think, in a lot of the talks that we've seen today, and certainly in the discussions we had with the scientists. And that is sort of Ron's motto. Figure out how to do the technology, and you'll just open up brand new possibilities in biology. So his approach to that and bringing together all these interdisciplinary people with diverse talents, encouraging them to collaborate, they're legendary throughout the field. It's what he's known for. And the fact that he's channeled all of that sort of expertise and vision and perspective into this disease is something that I think makes this community very lucky to have someone like him fighting for it. And Ron typically avoids the spotlight. That's kind of been his way as a scientist, but he's stepping into it now with a little bit of encouragement, or a lot of encouragement from his family. But I think we're all really lucky to have him and to work with him, and he's the reason that all of us are sitting here in this room today. So with that, I'm going to welcome Ron Davis to the podium.
So when Ashley tells you have to do it, you have to do it. This first slide is just as a reminder that all the work that we've done has been supported by the Open Medicine Foundation, and absolutely everything. And our first grant was the grant to do the T-cell activation, but that just got started. And and if you have to skip a talk, don't skip the next one. Because I think he has some really great ideas. And so I'm going to talk about something that is not that exciting, but something that has to be done. And that is, we really needed a biomarker. And so we've been working on that hard. And it's an extremely large need because of the fact that doctors say there's nothing wrong. And even some doctors say, well, if you don't have a diagnostic test, then the disease doesn't exist. And we don't have a real good diagnostic test. So we have to find one. So we've been working on this fairly hard. And just show you, this is just an update. We're getting there. There's a lot of work outside our lab as well. That's perfectly great. This is not a competition. And what we would do is to try to take the very best one out there and make sure it works. So we have a lot of experience in this area. Also, is to remind everybody, because doctors don't know this either usually, that a lot of diagnostic tests have a high rate of false negative and false positive. It's not uncommon to have a diagnostic test have 10% false positive and 10% false negative. So it's a real problem in the community because doctors believe it's 100% accurate, whatever it is. And if it's negative, then you don't have it, when you know for sure you do. So we have to solve that. So in fact, the National Bureau of Standards has established a lab at our Genome Center to really take on these kinds of problems. We've got to do better. We have to make it better for these diagnostic tests. So the simplest form of this, and Brain Hansen talked a little bit about this, is a commercial instrument called the Seahorse instrument. And this has potential, and I'm just sharing that it's not everything invented at our lab is a possibility. And we've gotten pretty good results with this if we use stimulated T cells. And what you see from this, and this is just the trace that you get out from the instrument, is that ME-CFS sample shows less activity than a healthy control, pretty reproducible. The instrument's a little expensive, although it can be multiple sample that could be put into a clinic diagnostic lab. But we'd like to try to do something that's maybe simpler and cheaper. But this is a little pilot study to show that there is a clear difference between them, reproducible. That's sufficient separation to probably make it diagnostic. And so and I'll talk about that a little later. This is an instrument that I have talked about before, but for those who haven't seen it, it's something that's been nano-fabricated. You can see on the upper right the size of this. This is using same technology used to make computer chips. And we have 2 gold electrodes that are very small. And they're separated. Silicon oxide is basically sand. It's not a good conductor. So and gold is a good electrical conductor. So this is just like a wire that you use to plug into something, except you can't see it because it's way too small. And then the other thing we do in trying to do better measurements is to take lots of measurements with the same instrument. A lot of technology takes one. And we take measurements from the 2,500 of these electrodes per centimeter, and we sample them around 200 times a second. So you get an extremely large data set in one experiment. That guards against just one false measurement. And then we use these multiple electrodes to measure electrical properties. Why we like electricity is that it's very cheap. Thank you. So if you do a test with blood in this device, healthy control just is unaffected. We put some salt into the blood. We don't know why that works, but we see after some time that there's a big increase in electrical impedance. So this is a little bit weird. It came about from studying some other things with cancer cells and so forth, which I won't get into. But the surprise was, in fact, it's very reproducible, and it's also very cheap. So it's a response to that ME-CFS cells somehow respond differently to salt. We got into this because we thought the cells had less capacity to make energy. And if you put in salt, the cells have to pump it out, and that takes energy. So they'll have less capacity to do that than a healthy cell. That may be what's happening, but we're not sure. But we also can maybe use this as a drug screen. We have now started doing some drug screens. We have found one drug that's that's used for other purposes that seems to have a big effect on this assay. We have to explore that a lot more before we talk about it. Now, the probability, well, let me just show you one. Here's a bunch of, we've done 20 samples with patients. The red is the patients, the green are the healthy controls. They're completely separated. And the probability that could occur by accident is 10 to the minus 9. So that's better than the lottery. And the chances that this is incorrect is very, very, very low. And so people often do the percentage of the likelihood of work. This is 100%. We've done a little bit of trying to understand where the signal comes from. We've done this plasma swap experiment, and most of the signal seems to be tracking with the plasma, which is something in the plasma that's creating the signal. Now that's encouraging because maybe we can figure out, we initially thought it was the cells, so we don't know what that is. And we've heard another talk about these small microRNAs. That's a candidate. That those small RNAs are causing this signal. Now we've also carried out a collaboration with San Jose State. Anand is an engineer, and he has built a microfluidic device that can look at blood. And this was an idea that patients have something wrong with their blood. So we looked at what's called the deformability of red blood cells. And you put them through a tiny little capillary, and but the red cells have to deform. This is just in your body. This is what happens in your body. They have to deform in your body to go through the capillaries. And red cells from CFS patients don't deform as well, and they have a hard time entering in this little capillary. And that's pretty reproducible. So what we have done now is looked at lots of different properties. There is a difference between immunosuppressed patients and healthy controls. It's not good enough to do as a biomarker because it's not completely separated. So we're building a new device that we think will give us complete separation. It'll be, again, a microfabricated little device. And then since that all got started, we've gotten to other individuals. It might even be here. I think Juan Santiago is here, and also Eric from chemical engineering. These are very high-level engineers. They do a lot of work for the Defense Department and so forth. Eric has access to Defense Department computers, so he's doing a lot of the modeling. And the plan is to build a different device to try out, which will be a handheld device that can measure this rigidity. One reason for having such a device and measuring it is that we could try, if it is a problem for the patients, we have an assay to try a diet change to see how we can improve that. Probably a lot of that rigidity is coming from the membrane, and it probably has to do with the oils that people are eating. So we can try out lots of different oils and see if we can improve it. So this could be another diagnostic instrument. It's also possible that we can combine them because these measure red cells and the other device measures white cells. We have another device that is, we can look at red or white, and it measures the magnetic levitation. And that's simply putting, the cells aren't magnetic, but if you put them in a fluid that is magnetic, then you can actually separate them by their density. And white cells and red cells will separate. We've been using it to separate out tumor cells that are in the blood supply. And here's just a picture where you can see you can separate different cells in this device. It's a little capillary, disposable. It costs 5 cents. Companies may not like that. But it could be made into an instrument because we see that this is the white cell population is generally light. and separates out from healthy cells. Healthy cells are on that reference line. So we may combine all of these into one instrument. That's a possibility. This instrument would separate the reds from the whites and then measure their density, and then it goes to the next device, which would measure the other properties. There are some interesting results here in the sense that there are a couple of samples that look weird, and it turns out they are happening right after a high fever. And after that high fever, the patient got a lot better. So for a short time, for a few months. And then we also did the plasma swapping experiments, and you can see a big effect there as well. And then this is looking at the instrument. So the instrument basically is a capillary, and all of the information is collected with a smartphone. So this is a new trend of using smartphones to build into the instruments, because it's a very powerful computer, and it's very cheap by comparison to putting in a real computer. So we're now doing what's called a bake-off, and that is we're taking blood samples and running them on all the instruments and looking to see how well they do. And this is also to see if we get any inconsistencies, and that will help us to looking at false positive, false negatives. And you're going to hear a talk, and also we're incorporating Bob Navio's. He came up with a pattern with the metabolomics that may be a diagnostic. We're going to put that in the system as well to see how well all of the different ones combined. And Robert Fair has come up with another one, which we can express as the tryptophan-kynurenine ratio or the activity of IDO1. And that also might be a diagnostic, and that's another thing that's easily measured. So you have to hear his talk to understand that one. But that's going to go into the comparisons as well. So the hope of this is we'll find some diagnostic tool that is very close to 100% accurate. It has to be able to be implemented, and it has to be simple enough that low-skilled technicians can run it without making big mistakes. That's not easy. But I think we have all the right experience and expertise to do that. So this is the team that we've had, and also here at Stanford, that have helped us in all this, and it is a big team effort. So thank you very much.